Welcome, everyone. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. We're really glad that all of you could join us today. I think this is going to be a really interesting webinar. Please feel free to put a note in the chat introducing yourself and where you're joining in from if you'd like to. I'm going to start with a few bits of housekeeping. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of environmental health sciences, the Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. CHE also hosts science discussion listservs and supports other collaborative programs focused on promoting environmental health and justice. If you're joining us by phone, you can download the slides from the webinar page on our website at healthandenvironment.org. And for those who are online, we'll put that link in the chat now. We will disable the chat during the presentations, but please do type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentations. And I'll just note there's a lot of scientific detail in today's webinar, so please don't hesitate to ask questions if there's anything that you'd like to have explained further. If you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. This webinar is being recorded, and it's scheduled to last for 45 minutes. With that, I will hand it over to today's moderator, Dr. Jerry Heindel, Director of Healthy Environment and Endocrine Disruptor Strategies, or HEEDS. Jerry?
Thank you, Rachel. And hi, everyone. Welcome to our webinar. Today, we're privileged to have Dr. William Goodson III present the results of his groundbreaking study on endocrine disrupting chemicals and breast cancer. Dr. Goodson graduated from Harvard Medical School and received his surgery training at Case Western Reserve and Boston Children's Hospital. He moved to California and was a professor of surgery at the University of California, San Francisco Medical School. Then he moved to California Pacific Medical Center, where he is a clinical research scientist at the Medical Center Research Institute. He is actively researching how environmental chemicals impact women's and children's health. He is also a spokesperson for the Halifax Project, which has raised awareness of the inadequacy of testing environmental chemicals individually instead of in mixtures. In 2018, he published another groundbreaking study showing that the mixture of BPA, methylparaben, and PFOA has effects 10 times higher than similar concentrations of the chemicals tested individually on breast cancer cells. He has over 30 years of experience diagnosing and caring for breast diseases. He was awarded the Compassionate Caring Award from the Institute for Health and Healing in San Francisco. He's a member of the Society for Surgical Oncology, the American College of Surgeons, the American Society of Breast Surgeons, and the American Society for Clinical Oncology. He has been listed in the Best Doctors in America since 1991. In addition to his practice and his research, he enjoys writing, having written 2 novels and a book entitled It's Your Body Ask, Your Guide to Talking with the Doctor About Breast Cancer. He also enjoys photography, biking, animation, road trips, and spending time with his family. All I can say about that is, wow, that is something. Today, he will discuss his most recent publication focusing on endocrine disrupting chemicals and breast cancer. Dr. Goodson, I turn the podium over to you.
Thank you, Jerry. It's always good to hear yourself introduced. Your mother would be very happy. I wonder about that. Anyway, it's a great honor to be here this morning. I am going to talk about our study And it's going to be more talking about the study than actually presenting the study. And I think as I go along, you'll get an idea of what I mean. That starts by pointing out that the title for the webinar, Reducing Breast Cancer Risk by Eliminating Parabens and Phthalates, would never be acceptable to a mainline medical journal, at least for the next 20 years. So what we have done, just a second, I got to make this thing go down. How do I make it advance? Crumb. Oh, here we go, sorry. The actual title is that reducing exposure to parabens and phthalates reverses accumulation of cancer-associated phenotypes within disease-free breast tissue of study subjects. It's much more scientific, and that's what you can get through reviewers. I would like to acknowledge the persons who have been very much a part of this. Shanaz Derki, by far and away, is the heavy lifter in this. She has worked with cells, and that's where I know her, and she's worked with cells for at least 40 years, of which I'm aware. Dan Moore is a statistician par excellence, and it's his statistical analysis that helped us salvage this study, as you will see in a while. Polly Marshall is our other co-investigator who is working with us on this project. For those of you who are interested, the article is open access. You can Google Chemosphere 2023, Dr. Derki, and it will take you to our article. So to begin with, we thank the many women who participated in this and the previous studies that are the basis for this work. This includes the 40 women, so approximately 40 women who were involved in this study, but the 240 women who were involved in the previous studies that helped us develop the way that this would be done. We appreciate the volunteers in breast cancer over time, which is our partner organization, and hereafter I will simply refer to us as we, rather than designating any specific participatory roles. We appreciate funding from the California Breast Cancer Research Program. They provided a small grant 15 years ago that got this project going, and they provided funding for the marginal costs of this study. The Clarence Heller Foundation, which helped us, and the hundreds of survivors and their families who have donated through the CPMC Foundation to develop the research team of Dr. Derki, Dr. Moore, and somewhat myself, making this and related projects possible. So we're going to talk about, how do I move that? Just a second. We're going to talk about gene expression. And gene expression is something that some of you are familiar with and some of you are not. So let's talk about that for just a second so that you know the term. You're going to see this slide or a variation on it 4 times in this talk. So please pay attention, and I hope I get it across to you. We talk about genes. Genes are our DNA, and the DNA is in the nucleus, shown here in the darker blue. We talk about it being in your DNA. That's almost a colloquial term. Those are the genes. But the genes don't do anything until they are expressed. And what being expressed by a gene means is that your nucleus, they make RNA. The RNA goes from the nucleus out to the cytoplasm, the lighter colored part of the cell, where the RNA goes to the ribosome, the little purple dots, and that is where new proteins are made. And the new proteins are what actually makes things happen. So measuring RNA, if you think about it, if you want to know what's going on in a cell, you need to know the proteins. You can tell what the proteins are going to be by looking at the RNA, and the RNA tells you what the cell is going to make and do. So measuring RNA tells us what genes are expressed. And in this study, we measured RNA levels before and after withdrawal of usual personal care products. I'll go through all this again, but I want to make sure you understand where we're headed. Personal care products change gene expression and thus change how cells function. That's where we're headed. So the takeaway from this study is that reduced exposures to phthalates and parabens from personal care products shifted the balance of gene expression away from ones that are associated with cancer and redirected the actual behavior of entire intact cells in the laboratory in ways that are more typical of benign cells and away from behavior that is typical of malignant cells. As I go through this, I want to point out, I'm going to hope you remember your high school algebra. This study is awash in double negatives. We reduce outcome, reduces genes, and that's positive. So you have a negative times a negative is a positive. And I will go through this several times, but just be aware that when we're talking about reducing and reducing, that actually may be a good thing. And we will talk about this more. So why is this study important? First of all, it addresses the problem of increasing breast cancer, particularly in young women. This study challenges the current assumptions about the safety assessment of chemicals. It demonstrates the need for a more comprehensive experimental database, and it provides and proves it's feasible to evaluate chemicals in evaluate chemicals in human volunteers. It complements but does not replace laboratory work, but it's an important step in sorting out exactly how things fit together. And it's possible. I think one of the things that really I hope to leave you as a message at the end is this study is a combination of laboratory people, statisticians, a clinical person, and the community people who help organize the volunteers. And most of the time when people are doing research, they tend to think, okay, I'm going to do research in the laboratory and I'm going to get a pathologist or a surgeon to flip me some specimens, but they don't think about reaching out into the community. So it's extremely important to recognize that working with the other parts of a team. So if anybody's talking about doing this kind of study, you can't do it on your own. You have to do it as a team. Now, breast cancer has been increasing since 1937. The first survey in the United States was in 1937. They found 65 cases of age-adjusted breast cancer per, it was 650 per million, which is basically 65 per 100,000. World War II got in the middle. They didn't repeat the surveys, but following after that, there was a gradual increase in breast cancer that starts after the war. This is the New York Tumor Registry, and this is the Connecticut Tumor Registry. This is the New York Tumor Registry. Both of them show a consistent upward trend. And that started a long time ago. It's very important to remember that it started between 1937 and 1947. And the reason that's important is because it's very common. You see a lot of people talk about the increase in breast cancer since 1973. And it is true, breast cancer has further increased from 1973, which is when the SEER data actually started. It peaked at about 2,000, just under 140 cases per 100,000 per year in 1999, dropped a little bit, and now is stable at around 130. It's actually 134 per 100,000 the last time I checked, which is around 2018. You can take PowerPoint, adjust your slides, and make it so you have the same time scale, 1980, 1970, 60, 50, 40, and make a continuous line. You can adjust it so that the 900 per million or 90 per 100,000 matches here. And all of a sudden you can see that this is a continuous line that goes starting before World War II, before my lifetime, and has been going up. Mammography may have had an effect, but mammography basically started in any large numbers in the 1980s. That's when the real campaign started. So you might attribute some increase to mammography, but there's a lot of increase before mammography came on the scene. You also can point to hormone replacement therapy a little bit here, but as I'll show you, there's a lot of effect in younger women. What does happen here is DDT, DES, polyethylene terephthalate, or the plastic made from BPA, and a whole bunch of other things that are going on here that tend to be ignored. And it's because this is an easy graph to draw. What I chuckle at is if you go to SEER data and cut and copy and paste the data from SEER into an Excel file and simply make a chart of it, it actually comes up with this same coloring almost every time. You know what people are doing, and it's a very easy thing to do. But it is incomplete. The whole picture goes back a long way. And just to back this up, this is a recent study from a woman who, and I can't see her name because of the way the little bar is on my screen, but she was at the University of Kansas, has now gone to the Mayo Clinic. She looked at SEER data and showed that the annual incidence of breast cancer in women under 40, 39 and younger, these women have no screening, no hormone replacement therapy, has gone up at about a 0.5%, average annual percentage increase of about 0.5% a year. on average constantly from 2004 to 2017. Breast cancer in younger women and breast cancer across the board is increasing. And you have to look for other reasons because delayed childbirth and oral contraceptives have been around a lot longer than starting in 2000. Those are really issues that came up in the 70s and 80s. You can't say that's the explanation. Increase in breast cancer began 30 years before mammograms. And I don't have the slide here. If somebody wants it, I can show it. People who have BRCA carriers, every generation, the younger generation gets breast cancer an average of 8 years earlier than the older generation. And the increase in women under 40 years of old cannot be explained by screening mammograms. That just isn't part of the group of people, even when the new guidelines start at age 40. So what are suspects in this? And we've focused on parabens and phthalates. They're ubiquitous suspects. They are common in personal care products. You know, unless you have something that is specifically free of parabens or phthalates and fragrance, as we'll talk in a minute, you're probably, if you've gone down to your drugstore and bought something on your shelf and used that this morning, you probably put it on yourself. Phthalates and paraben excretion correlates with personal care use. So it's like drug testing. You know, people talk about somebody gets a drug test. We probably have at least heard of that. You can monitor exposure to phthalate and paraben, and it correlates with PCP use. This is a variety of studies done by, there's a study where they were decreasing it, and we'll talk about that in a minute, but there are other people who simply correlated use. So it clearly is associated with personal care product use. There are also other xenoestrogens that we're exposed to. These are things that come from food packaging, house dust, fireproofing, stain-resistant materials. If you have a carpet in your house that is stain-resistant, it probably has PFOA on it or something like that. And little tiny bits are out in the dust all the time. So we're exposed to these all the time, but we look specifically at phthalates and parabens. That was our interest. Now, the background for this study, there was a lot of people who were building on their shoulders. Probably aside from the interest in phthalates and parabens, what they'll do in the laboratory, Philippa Darbra at Reading in the United Kingdom did a landmark study where she got a hold of tissue from mastectomy specimens, women having their breast removed for treatment of breast cancer, and worked with a biochemist to measure phthalates and parabens in human breast tissue. It is there. But not only did she do that, what she did that was even more important is she took the combination and the levels, the concentration of these xenoestrogens in actually measured in human breast tissue and exposed cancer cells to it. And the cancer cells really kind of went wild. MCF7 is Michigan Cancer Foundation number 7. It's a very commonly used cancer cell that is responsive to estrogen. So that's very important. The second predecessor here is Carol Fabian and Seema Khan. Carol Fabian at Kansas University, or KU as we call it from Kansas City, Seema Khan at Northwestern. They set up study design where they did RPFNA, as we'll talk about a little bit more in a moment, before and after an intervention. So the idea of doing a FNA and then doing an intervention and then doing an FNA again is something that they pioneered, and I need to give them credit for that. Next is that the background is that Dr. Derkey has led our group for the last, it's now 15 years, and the study started, we had 10 years of experience analyzing fine needle aspirates. You have extremely tiny specimens. I always refer to the specimen when we talk to a patient about getting it. It's about the same as less than a reverse flu shot. You're never going to notice the difference, but she has worked out ways to work with this. And then the thing that really brought this all into focus was the Hermosa study, a small study, 3 days, high school girls in Salinas, California. All they did is got urine samples, gave them 3 days of cosmetics that did not have phthalates or parabens, and got another urine sample and showed that you could have a marked reduction in the xenoestrogens for just 3 days. So it was logical to try to figure out what can you do if you do it longer, and can you measure a difference based on, we know it's in the breast, We know that there's a good study design before and after. We've learned how to work with these cells. We know that you can change it. Is there a way to measure that effect? So what we came up with is what we referred to as the reduced xenoestrogen exposure, abbreviated as REDUX here. And we screened volunteers to determine whether they used at least one product every day that contained a phthalate or paraben. And as I said, if it was scented, we considered that as a surrogate because the law requires companies to list their components in their over-the-counter products, but fragrance is considered proprietary. And a lot of the use of phthalates and parabens is considered or shielded as being part of the fragrance, so that they deny that that's there because they don't have to put that in. California has legislation that I think will go into effect in the next couple of years that will require listing that, but that, as far as I know, is the only place that's been enacted. We designed a crossover trial. We wanted everybody to get 30 days of intervention, but we had people who did it first and people who did it second. And this was 28 days from first sampling to 28 days of the second sampling. We recruited women who had relatively regular menstrual cycles who were on birth control pills. And we assumed that about 70% of the cells would grow, and that if we randomized 60 people, we would get enough to make sense of it. The intervention provided subjects with a 30-day supply of phthalate and paraben-free personal care products identified by searching the Environmental Working Group website, ewg.org. They have an entire website that tells you what's free of things. The key thing is to get stuff that don't have fragrances, but we'll go from there. You can go there for yourself. We measured the effects of the REDUX with random periareolar fine needle aspiration. Fine needle aspiration means a little tiny needle about the size of what you get your flu shot with. Random means that you don't have a target. You're just putting it in the breast tissue in a specific area. We do it around the periareolar because that is the area where breast tissue tends to focus. And it's a needle biopsy using a very small needle. We obtain samples before and after the intervention. We use local anesthesia, and I always point this out to people. For the naysayers, we had no attrition. Everybody came back. You can anesthetize the breast and make it so that this is acceptable. We did cytology with a pathologist who confirmed that there were no atypical or cancerous cells. We weren't accidentally stumbling onto something. And this is where Dr. Derke and her wonderful hands came in. We expanded the cells to have sufficient numbers to measure RNA and cell behavior. There is no technology right now that will allow us to do it straight on the FNA. Actually, it wasn't available at the time. There may be technology now, but the world is changing very rapidly, and the ability to measure RNA on individual cells as they come out may be available soon. So we wanted to know whether or not women, the volunteers, actually were using the materials that we gave them. And we did this by doing urine and blood testing before and after the intervention. As I said, you can tell what things people are exposed to by urine testing. And we compared the samples, and there are two things that are important in here. Only the subjects who were in the intervention had a significant decrease in 4 parabens that are associated with cosmetics, methyl, ethyl, propyl, and butyl, and 1 cosmetic-associated phthalate metabolism. It's MEHP is a metabolite of DEHP. That's monoethyl phthalate is what you get when you remove a group from the diethyl hexyl phthalate, which is common in cosmetics. Those things decreased. However, we also looked at a variety of other common phthalates that you're exposed to from food and other sources, and we found no evidence that volunteers changed other aspects of their lifestyle. So we feel that this is strong evidence that what they did is change what they use for cosmetics. Polly is going to talk in a little while and Ms. Marshall can comment on the acceptability of these products to patients, to the volunteers, because my understanding is that they were really quite acceptable, except for the nail polish. Apparently it chipped rather easily. We also looked at estrogen, progesterone, sex hormone binding to see whether there was any change. So we got this project underway, and along came COVID. And that's a lot of what I'm going to talk about here, because it's sort of figured out how Dan Moore, our statistician, was able to make sense of it anyway. We started accrual in the late 2019 with a planned 60 volunteer subjects. In February and March, we had some soul searching and realized we could not ask people to come to a research center in the middle of COVID simply for us to do a research project. At that time, we had 14 successful pairs. Recall that if you do the first one and the cells don't grow, it's not worthwhile doing the second one, because you don't have a comparison point. But we had 14 pairs. Unfortunately, it was skewed 12-2 in favor of the intervention. And this led to some significant problems, and led us also to realize that we needed to work, we were planning to work with Dr. Moore, but we also needed to really work with Dr. Moore. He needed to work with this a lot for us. And that, my hat's off to him, because he did a great job. What we also did is we were able to validate the changes in the genes by looking at what happens in whole cells. And that is a very important part of this. So remember that we measured gene expression. We determined which genes were expressed differently between the controls and the intervention subject. This is sometimes said differentially, that's science speak, but what they mean is there's a difference between how the genes change between the first and the second time. Now, I'm going to have two slides that are somewhat technical. You can ignore them or try to follow along, but I'm going to come back to more typical stuff in just a moment. But just so if anybody's interested, what we did is for all 14 subjects, or what Dr. Moore did, all 14 subjects, we had a measure of RNA or gene expression in the first sample and the second specimen. We calculated the ratio of the second one versus the first one, so that you simply divide the second one by the first one, and for all 566 genes, and we took log 2 of this. And what this means is if RPFNA was bigger than this, if the gene was more expressed in the second one, the ratio was greater than 1. And since the log of 1 is 0, if it was greater than 1, then the log of 2, log base 2 was greater than 2. And this is a very common practice in doing gene analysis. You convert everything to log base 2 because it makes the outliers less likely to skew your data. And then what we did is we compared the log ratios. There's always some drift between the first one and the second one. And so we compared what happened in the intervention subjects to what happened in the control subjects for each gene. We had 566 genes. Again, we used a p-value of 0.01, which again is a very commonly accepted number. And by chance, if you were going to take 1 out of 100, you would expect 6 genes by random chance to be, quote, statistically significant. But we found 64 genes that were more common. So we felt that this was very much what we wanted and what we were interested in. Oops, back, I lost the slide there. But as I said, it led to some questions and the skewing. We had to sort of work with this. So Dr. Moore used other processes. One is leave out, random leave out, just went through on each gene, randomly took one out to see whether or not it was still significant. And by randomly removing genes, we lost some, and we are no longer subject to being significant. But the more important part of it is we basically took this is done for each set of gene profiles for all 567 genes. And we randomly selected and say, okay, what happens if we just randomly select the controls and just take any 2? And could by chance, you take these 14 and pick 2 out and call them controls, could you by chance get situations in which it would look like it was a significant difference? And that would happen some of the time, but there were only kept the genes where you couldn't get a random rearrangement that made a significant difference. And that was 26 genes. So those 26 genes are the ones that we focused on. It's very important in looking at that to realize that 18 genes decreased relative to controls and 8 genes increased relative to controls. There's a very important concept in that, and that is that getting to and from cancer is not you have one gene that gets more, you get more cancer, and one gene that gets less, you get less cancer. There's always a balance. Some genes cause cancer, some genes oppose cancer, and you need to figure out how that works. And of the 26 genes, 18 of them shifted in a way that we would think more normal, but it also means that 8 genes shifted in a way that might actually, you'd think, promote cancer. So just looking at it overall, and I hope you guys can see the bottom. On my view, I lose the very bottom of the slide. I'm sorry for that. But we had 18 genes where the expression decreased compared to controls. And of those, 14 would be ones where being lower was better, but 5 of them were ones where we really would have preferred the gene to go up. These are genes that tend to suppress cancer, and you would like them to go up. So when they go down, it kind of works against you. Likewise, we had 8 genes that increased. 4 of them we would like higher was better. That was good. But 3 of them, we would prefer it to have been lower, so going higher, particularly NRAS, which is a big gene, not in breast disease, but a big gene in melanoma, for example. You would prefer that to be lower. And the had one gene that's a little hard to know what it is, so I counted it on the side where you weren't happy with it, but I kind of put it in yellow rather than saying it was absolutely red. So how do you make sense of that? First of all, you can use programs, and this is open access programs, this is in the paper, and I would point out that I'm talking about the paper, but the paper is open access and you can download it for yourself. We looked at the various pathways that were significantly involved, P less than 0.05 and less, and the PI3K AKT signaling pathway is a major pathway in cancer, as is phosphoautophagy. MicroRNAs are a big part of cancer. mTOR signaling is sort of downstream for the PI3 AKT pathway, and it's a major focus. It's almost like the hub, some people say, of cancer. Herb signaling is always important. So these pathways are involved, and let's to sort of figure or give you an illustration of how this works. Let's go through one pathway, and the PI3K pathway is very important. This is the pathway of genes that when PI3K is activated, it kicks off AKT, AKT kicks off mTOR, kicks off a couple more genes and goes to the cyclins, which makes cells grow. And this is a central pathway. If you look in the second column, at the top part here, there are 5 genes that would typically promote cancer. And then down at the bottom, there's 1 gene that drives cancer and 1 gene that suppresses cancer. And 1 gene, as I said, we don't quite know. That's where the COL4A5 comes in. These 5 genes that promoted cancer also had reduced expression. So reducing expression of a gene that promotes cancer is a good thing. These genes, these 3 genes, remember there are 8 in the group, these 3 genes had increased expression. DDIT4 is a cancer suppressor, so increasing the suppressor is a good thing. But NRAS, it drives breast cancer somewhat, but it really is particularly common in melanoma. The skin cancer, and COL4A5 is a protease that might help cancer invade, but we don't know for sure, but I put it in that, I just mark it out by itself. So what you can see is you have a combination of, you got more things that are good, and actually you got 6 of 8 are good, 1 that's definitely bad. How does this all balance out? And how do we know the overall effects of the balance are good or bad in changes in the gene expression? Because I just showed you we had 5 good and 3 bad. And what we do is we evaluate the behavior of intact whole cells. And fortunately, and this is where Dr. Derkey really gets the prize, she has figured out ways, we not only grew little bits of this, but was very successful in growing cells. So that we had some live cells from before and after the intervention that we could then study in the laboratory. we looked at several things. I'm going to talk to you about estrogen receptors. There are two estrogen receptors, alpha and beta. And when somebody talks about an estrogen receptor positive cancer, it usually means estrogen receptor alpha has gone up. ESR2 is how it's also sometimes called. ER beta is actually typically found in benign tissue, and it's lost in the transition to malignancy. And what we've previously showed in our laboratory studies, which we'll put on the webpage later, is that benign breast cells exposed to xenoestrogens lose ER beta. We know that in the laboratory, and now we can show it here. So what you have here is data from two subjects. That's the first set, second set, third set, and the second patient, second patient, looking at estrogen receptor alpha, the amount of the actual protein. Remember, when a gene is expressed, you get the protein, the phosphorylated or the activated estrogen receptor alpha and ER beta. And this is the sample rPFNA1 and rPFNA2. The green bars are stuff that I'm not going to get into right now. It's the in vitro exposure to estrogens in the laboratory. That also changed it, but that would make this a 2-hour talk. What you can see is that before and after, the levels of estrogen receptor alpha in the cells went down. And that is shifting away from the behavior that you would see in cancer. The levels of activated estrogen receptor went down, before and after went down, before and after went down. And what is really very interesting to us, and which really kind of confirms this, is the levels of estrogen receptor beta went up. Remember that estrogen receptor beta is more typical of benign tissue. And one of the things, you can look at breast biopsies and measure ER beta levels. And when the ER beta level goes down, and then you follow these patients, you will find cancer is more common when the ER beta is down in benign tissue. So we feel this is very exciting to us. And it's a confirmation in whole cells. We also had sufficient samples from 3 patients to measure apoptosis. And apoptosis is sometimes called programmed cell death. If you've heard about immune therapy for cancer in the last couple of years, it's a way to recover the apoptosis program in cancer cells. That's what kept Jimmy Carter alive since the 90s. Normal breast cells self-destruct when you trigger programmed cell death with tamoxifen. Loss of apoptosis is a hallmark of cancer, and benign cells exposed to xenoestrogens in the laboratory lose apoptosis. And you can see that the apoptosis is higher after the intervention, higher. Now I'm really running over time, so I'm going to just wrap up here real quickly. Personal products, reduced exposure can change gene expression, can change cell function. We're very proud of this study, but there are some limitations. There are small numbers, skewed randomization, however, intact whole cells support what we find in the gene expression. It needs reputation. And like other gene expression studies, you can't really, except for things like a few things like P10 mutations, P53, BRC, you can't tell from a mutation what's going to happen to cell behavior. We need much more data on that. And the benefit of this study, however, is it is a randomized trial. We showed the effect on gene expression in whole cells. And currently, this is the only human study out there, and we found that chemicals in personal care products adversely affect breast tissue, and that is not what anyone would want. I apologize for talking so long. Thank you very much.
Okay, thank you, Bill. That was terrific, and it was very well done and organized very nicely. So before I turn over to questions, I'm going to turn the podium over to Polly Marshall. So she founded the Breast Cancer Over Time, a nonprofit created and controlled by breast cancer survivors and dedicated to supporting scientific research into breast cancer prevention. She was the community principal investigator on this study, and she's going to present an overview of their participation in the study and their view of the results. So, okay, Polly, go ahead.
Thank you. I am going to speak relatively quickly because there's not that much time, but I'll let you know I'm Polly Marshall. I am a 13-year breast cancer survivor. These are the women who started my organization, Breast Cancer Over Time, back in 2015. We're all breast cancer survivors, and we were all members of the same support group. To give you the context for this, we, between us, had months and months of chemotherapy, of surgery, of radiation, of hormone treatments, and we've been living with breast cancer for years and years and years. But importantly, we have, most of us have children, and we have sons and daughters who are at higher risk of breast cancer simply because they are our sons and daughters. So speaking of our daughters, when members of our group emerged from our own treatment and started to be able to look at what's going on in the rest of the world again, our first thoughts were towards our children. What could we do to prevent our children, and then by extension their friends and the whole next generation, from going through what we have had to go through? We wanted to know answers quickly, like what risks do our children face that pose the greatest danger? What exposures create the greatest danger? And most studies that are on what causes breast cancer involve, for obvious reasons, involve correlations. These exposures here led to breast cancer 10, 20, 30 years later. We had a sense of urgency that we wanted to know faster. So this is our team, our research team. Dr. Goodson was my surgeon. It turns out, he was my surgeon, and I found out about his research because he actually requested that I donate cells from my uninvolved breast when I was having my lumpectomy surgery back in 2009. He introduced me to Dr. Derkey, who he had been working with for decades on this science of what exposures do, and particularly what mixtures of exposures, of chemical exposures, do to human breast cells. Dr. Derkey explained to me that most research, like cancer research, is done on rodents and on cancer cell lines, and that it's actually much more efficient to look, if you want to study prevention in humans, which is what we wanted to do, the most efficient thing is to have actual human breast cells to work with. So you've heard about the Hermosa study. I just wanted to show you what some of these girls look like. But this was our inspiration. This was a study that showed that if you took cosmetic chemicals away from these girls for just 3 days, the content of these xenoestrogens in their urine dramatically decreased. So that was our inspiration. From there, we went to who we could recruit. And my daughter is actually in this picture. But we knew that we could get young women to donate breast cells. And indeed, my daughter got every member of her college soccer and basketball teams, because she played on both, to sign a letter saying, we would donate cells. We want to do something real. We're done. We're sick of awareness. As Dr. Goodson said, we looked at the contents of what people were using to qualify for them for the study. And importantly, we shared with them, what is it that you used that qualified you for this study? And so we're very proud, and we think it's an important part of community-based research to actually educate the participants. And again, these participants came back for their second fine needle aspiration, every single one. And we think it's because they've all been affected by breast cancer. They did this in honor of their mothers and their aunts and their grandmas. They did it for their generation. And we, the survivors, held the hands of every single one of them both times. They had needles in their breast to donate donate tissue samples. So we had several take-home messages to our participants and indeed to their whole generation. You need to pay attention to what's in your products. There's all kinds of ways you can look it up, and there are healthy products coming out every day. Be aware that the FDA does not regulate cosmetics, and so nobody is looking out for you. However, the state of California will ban certain ingredients starting in 2025. And I'll just mention that both Dr. Goodson and I were up there in Sacramento testifying to help move this legislation forward. Two other quickly messages. One was to scientists. Please take notice of this research. This methodology could be used to study other kinds of exposures. And to the government, please use this data to regulate cosmetics. It's been done in the EU, it's been done in Canada and in Japan, not this research, but actually banning certain chemicals in cosmetics. So I will stop there. Thank you.
Okay, thank you very much. That was very, very nice addition. So I wanted to ask a kind of a question to both of you, starting maybe with Bill. So to my mind, this is one of the most important kind of studies out there, something that really will help link chemicals to breast cancer. So we need many more of these, and how are we going to stimulate NIH and other organizations to fund these kind of research? I know, Bill, for example, you're a member of a lot of very big, important societies. Do they ever get together and say, hey, this chemicals in breast cancer is important, and what are we going to do? And here's an example, and try to push this kind of research?
I think I'm reminded of the guy in Greek mythologies always pushing the rock up the hill. I think that's there is a tremendous amount of inertia. I will relate to you a conversation and an experience. One is in 2019, the American Association of AACR, American Association of Cancer Research, had their first ever seminar on the effects of chemicals and possible relationship to cancer. I spoke at that meeting, and they were supposed to put together a a special issue of the journal Carcinogenesis afterwards. And my paper was rejected because I talked too much about chemicals. Everybody else was kind of talking about stuff around chemicals. And I particularly included the idea that breast cancer is increasing, and the dogma is that it's all because of mammograms. We need to get the message out. The same person who says it's all overdiagnosis was Richard Peto, who's the same person who wrote the paper in 1980 and said it was increasing. So you can't have an overdiagnosis between 1950 and 1980 and then overdiagnosis again. You got too many people dying. The second thing is that I had a conversation, I won't embarrass this person by name, but a previous president of ASCO had a conversation about 5 years ago, and this person and I were talking about it, and I said, why is there no research on this? And his comment was, it's just not what people are interested in. It's not mainstream. And I didn't want to be offensive, so I kind of didn't push it. I didn't want him angry at me because I still kind of like wanted to work with him. And I said to him, I said, okay. And then as we were going our separate ways, he said, Bill, you're probably right, but you're never going to prove it. And so I think that this study, what we've been able to do cooperatively, is something that needs to be tossed out to the people and who are making this stuff happen. I mean, somebody can chew at this study and show you all the things that are wrong with it, and I know that. That's why I've gone through that. But the bottom line is that even when you go through all the things that are wrong with the study, you don't like the answer. And I think that we need to recognize that this is going to take change. And in the answer to your question, there's no easy way to do it. I gave you examples of why it's so hard.
Holly, would you like to say something?
Yes. Well, the study was funded by the California Breast Cancer Research Program, which is cigarette tax money. And I think that's very significant. They fund this kind of research. They're interested in the environmental exposure aspect of it. But it's also true that it's very hard to get money to fund research into prevention. And why might that be?
Nobody makes money.
Not to get too political here, but there's money to be made on the cure, which anybody who knows a little bit of science knows that the cure is something that really will never exist. There is no money to be made by hospitals or drug companies on prevention, because all that does is, it saves our lives and saves our breasts. So that's what we're running up against. But I think that this study shows why I showed you the pictures of the people who were involved in contributing cells is because there is passion out there from young people. There's passion out there from environmentalists. Those are the people who trooped up to Sacramento to talk about this stuff. And when you put the human face on it, which we're able to do in this collaboration, it gets you further. But there's a long way to go. And we're just so happy that we're able to put together the scientists who have already been working on this and figure out how we could support them. And that's really what this was all about.
Okay, thank you. So there's one question I know you can't answer, but it's kind of provocative. Since breast cancer has been going up now long enough that it's over generations, and some of these chemicals like phthalates have been shown to have transgenerational effects. Maybe some of this is just increasing because the grandmother was exposed, and now there's increase in the daughters and the granddaughters, and that's going to be hard to tackle.
I think that's hard to tackle, but I'm going to step away from phthalates and parabens and go to diethylstilbestrol. DES is the poster child, so to speak, although I'm not sure you want to call it a child, the poster devil of environmental chemicals. And we know that DES was found to cause cancer in young women in the 60s and was sort of stopped used in the very early 70s. But we're reaping the side effects of that in the 80s and the 90s and even in the 2000s. And, it's the gift that keeps on giving, as people like to say. And we don't know what the long-term effects are going to be. There are studies that look not only at effects on cancer, but there are studies looking at levels of these chemicals in women during pregnancy and showing effects on their kids, or their little boys. The little girls are less affected because they're supposed to have estrogen. Little boys are not supposed to have estrogens. And I think that you've asked the bingo question of what's going to go on in the next couple of generations, because there are, the funny thing is that there are some generational changes that do not appear in the first generation male, but only shows up in the second generation offspring of the first generation male. So mommy's exposed Little boy looks fine. Children of that boy, now this is laboratory animals, but you know, we are all laboratory animals in this giant experiment. We hear about climate change, and I don't know which is worse. I don't know whether it's worse that we're going to be swimming or whether it's worse that we're not going to have kids because we're all sterile. That's really what I wrote a novel about. So I think that's you know, we need to think that through.
There's one question that's very scientific. How was the expression of apoptosis mediated? Was it changes in expression of CL2 family proteins?
It's actually an exon, which is expressed by cells in apoptosis and with a cell sorter. And the methods are in the paper. The paper is free online, but we exposed the cells to tamoxifen and then looked at what they would do. It's a provocative test. We've used this in with BPA, methylparaben, terephthalic acid, PFOA, and the mixtures, as you alluded to before.
Okay, well, thank you, Bill and Polly. This has been absolutely phenomenal. Appreciate your time. And now I'm going to turn it back to Rachel to end the webinar.
Thank you, Jerry, and thank you so much, Dr. Goodson and Ms. Marshall. Thank you so much for this amazing research that you have been doing and for such excellent presentations. And I just wish that we had more time to discuss all the additional questions that people have. But just to wrap up, just to let you know that a video recording will be available on CHE's website soon, and everyone who registered for today's session will receive an email with a link to the video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's Partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners. We also invite you to join us for upcoming webinars. So we have a really interesting one coming up next week. On Tuesday, May 23rd at 11 a.m. Pacific, 2 p.m. Eastern, we'll be highlighting the work of Dr. David Carpenter, Dr. Tyrone Hayes, and Stacy Malkin in Industry Influence and Scientists, Protecting Scientific Integrity. This webinar will look at common tactics used by the chemical industry to pressure scientists and explore how leading researchers have maintained their scientific integrity in the face of industry pressure. And that webinar is brought to you in partnership with the Program on Reproductive Health and the Environment's Science Action Network. And in just a minute, I will put that link into the chat. So again, I'd like to thank all of you for joining us today. We do hope that you found this information to be useful, and we look forward to seeing you on future webinars. Thanks again so much to our speakers and our moderator, and thanks also to CHE Director Kristen Schaefer for all of her behind-the-scenes support. Thanks again.